right, good evening. Thank you everyone for being here. We are delighted to invite you all to the Helen, Marcy, Golde breast cancer lecture this year. We're so excited to be back in person and virtual after a hiatus last year, and just delighted to welcome our speaker and all of you to what we hope is a really lovely evening to learn a little bit of science and to honor the memory of Helen Marcy Golde. So Helen was a marine scientist who spent much of her career at the National Oceanic and Atmospheric Administration, and in 2008 developed breast cancer that was prompted by the BRCA2 gene. Helen did an amazing, participated in clinical research. She gave so many gifts throughout her course, and we're honored with being able to continue to learn more and to think about how we can manage and make breast cancer a more successful treatment for many other women and other individuals who are to come. So by way of introduction, my name is Jennifer Specht. I am an associate professor and I am the clinical research director for the breast oncology program at the University of Washington. And I'm really excited to be here with my friend and colleague, Dr. Hannah Linden, and we are honored to welcome the Golde family this evening. We are a team of breast oncologists. We have a multidisciplinary team, and it's one of the most amazing things about working here is that we have the opportunity to work with clinicians, we work with scientists. Dr. Linden and I, and you'll see Dr. Mankoff have a deep interest in how we can use molecular imaging and other tools to help us do better for our breast cancer patients. And so it's wonderful to have some of our colleagues join us here this evening as well, and to those online also. With regards to just some logistics tonight, we are here in person, but we're also trying to make sure that those of us who are joining virtually have an opportunity to participate and to hear Dr. Mankoff's lecture. And so for those of you who are joining virtually, you should have the ability to add questions on the chat function, and Dr. Linden will be monitoring that, and we'll try to make sure that those questions come to Dr. Mankoff at the end of his talk so we can make sure to answer all of those. So in terms of next steps, I'd like to introduce Helen Golde's sister, Chris, to say a few words about Helen and to continue with the introduction this evening. So thanks very much, Chris.
Thank you. I can wrestle off my mask here. I've gotten good at wearing masks, but not very good at taking them off in public places. Very exciting. So good evening. My name is Chris Golde, and on behalf of the Golde family, first we want to thank Dr. Linden and Dr. Speck and Doreen, whose last name eludes me in this moment, who did all of the work to help make this lovely event happen, and of course for Dr. Mankoff for joining us here today. And we're glad that all of you around the country can join us for this event as well, or if you're watching the recording later. Every year, we're really delighted that the Helen Golde Lectureship brings a noted breast cancer researcher to Seattle to interact with the students and the faculty at the University of Washington, the Cancer Center, and the Fred Hutch, now the umbrella organization, to really advance science and to share the latest on breast cancer research. The speaker always does that in consultation and in seminar format on a separate day, but we are also delighted that it's kind of a two-part event, and we get to have a public lecture as well. This public lecture honors my late sister, Helen, who died of metastatic breast cancer in May 2015. She was a scientist who embraced clinical trials in her treatment and really would have enjoyed this event. Today would have been, well, not today, but we always pick the month of May because she was born in the month of May, she passed in the month of May, and so she just recently would have had her 59th birthday. You can follow the link we put on the slide, in the chat, on the Zoom link, and in the brief bio that we sent. All of that is the same text of a brief bio of my sister. So we are excited to be back in person so we can, hug and elbow bump here. And now it's my privilege to introduce you to Dr. Mankoff. Dr. David Mankoff is the Matthew J. Wilson Professor of Radiology and Vice Chair for Research in the Department of Radiology, as well as the Associate Director of Education and Training at the Abramson Cancer Center, all of which is at the University of Pennsylvania. He practices nuclear medicine with a special interest in oncological applications of molecular imaging and radionuclide therapy. This is a pronunciation challenge here. His research focuses on molecular imaging of cancer, primarily on breast cancer, and emphasizes the therapeutic monitoring identifying factors mediating therapeutic resistance, and the translation of new methods into clinical trials. He also works on imaging methodology and quantitative imaging methods related to molecular cancer imaging, and he's promised to show us some gizmos tonight. So, without further ado, Dr. Mankoff.
Thank you very much, Chris, and I very much enjoyed talking to you and your family about Helen. Sounds like a remarkable person. And I want to thank you for supporting this wonderful breast oncology program and the ability to come back. It's been 10 years since I've come back in some official format, and I'm really excited to be here and see so many old friends from very different venues, mentors, and so it's really great to be back here. I am going to tell you a story of work that was really, a lot of it was done here. I was talking to Hannah and Jen this morning and telling her that I'm at Penn, and people go, aren't you supposed to start publishing with the people from your own institution, not your former colleagues at the University of Washington? And so as I put in the acknowledgments, I don't know how well it comes across on the screen, I tried to put purple and gold for all the people that have contributed to this that are either here or were here. Some of them are now at the University of Pennsylvania and other places. So I'm going to talk to you today about molecular imaging for precision breast oncology. I'm going to talk to you about steroids again with a lot of the idea that that work was done here. And then I'll talk to you about some more recent work with PARP imaging, which is quite relevant to the BRCA gene mutations. I am conflicted. I have all the things in yellow are things I'm going to talk about today, including consulting arrangement with GE Healthcare. I will tell you that the PARP stuff that I'm going to show you tonight is a company that my wife is the CEO of now, so I am conflicted, and I want to make sure I declare that up front in this talk. So I'm going to talk to you today about PET in the general format as a tool for precision oncology, using imaging to guide hormone receptor targeted therapy, the estrogen receptor. Some relatively more recent and a short introduction to what we're doing in PARP targeted therapy, and as Chris mentioned, some imaging tools. I did start life as an instrumentation physicist. So for those that don't live, eat, and breathe PET scanning, this is what happens in a PET scanner. Each morning we fire up the cyclotron that is sitting below our nursing building at Penn. And it makes a radioisotope that's usually short-lived, in this case fluorine-18, that has a 2-hour half-life. We take it to this gleaming GMP facility, good manufacturing process facility, to make a radiopharmaceutical. I'm illustrating a glucose analog, fluorodeoxyglucose. I'm also illustrating why I should not be a medical illustrator, and I should probably keep my day job. But the patient is smiling, something hot and red is being injected, and we put them in a scanner, which these days is a PET scanner, typically with a CT, sometimes an MRI. And what we get out of that is an image of tracer concentration. That has two important meanings. It's an image, so we can see what's where and take pictures and use this to detect and characterize cancers. But it's an image of quantitative concentration, so it gives us numbers that will help us not only see these things, but to be able to use them to direct therapy, to assess how well the therapy is doing, in general, to gain some insights into how the tumor is behaving. So I would argue that for the most part, we are still largely using this technique to detect cancers. So here's an illustration of fluorodeoxyglucose that leverages the fact that cancers tend to have an elevated and aberrant metabolism of glucose. And this is a woman who has right locally advanced breast cancer. In my image is usually black means hot, that's more radioactivity. But had otherwise undetected spread to the mediastinal nodes, which was easily detected by PET and was important in help guiding treatment. There have been other tracers that have been used. If you tune in tomorrow, I'll talk to you a little bit about the biology of fluciclovine. It's an amino acid that has been used, for example, to image prostate and brain tumors. But these have been very successful in the clinic and have been used largely to identify cancers and to stage them and find out how far they've spread. But today I'm going to talk to you about something different and really using these to be able to characterize tumors. And to be able to look at the types of targets that might be amenable to them, and to really guide therapy, not just find where the tumor is. And I'm going to concentrate today on nuclear receptors that are part of a basic female hormone therapy, estradiol or the estrogen receptor. And I'm going to talk to you at the end about DNA repair and PARP as well. Very importantly here, it is not just looking for a hotspot somewhere. It's how much of this particular target is present and how much that target is changed. And in particular, the absence of that target, if you're trying to direct treatment there, may be as important as its presence. And this is a real departure from the way we've used imaging for a while. And the first example that I'm going to show you about estrogen receptors, I think is really the first example we've seen make it into the clinic and be approved to not only detect cancers, but guide their treatment. Okay, here's your chemistry question for the day. Why do we do something crazy? Why do we take this short-lived radioisotope, go through this complex chemistry, make it under really strict conditions by the FDA, inject it into a patient, and take this very specialized scan? And the reason for that is the high contrast it has. Most of you, I assume, right now are not radioactive. So if we make you a little radioactive and put you in the scanner, we can detect, in theory, just about every molecule that was injected. I'll show you some devices that help us with that in a second. So in units that may not mean something that much to the folks that are nuclear medicine, we can make a dose with about 5 millicuries of tracer. And when I go through all the calculations, we can do that with injecting nanomoles, 1/1000th of a mole, and get down to very low concentrations. And these concentrations are absolutely important for these very sensitive receptors, where if you gave too much of the material that you were trying to image, it would be the imaging Heisenberg uncertainty principle. You wouldn't know what you were looking at. You were perturbing it. So these PET tracers can image processes that are very sensitive without disturbing it. And pretty much any other technique that you would use right now in imaging is going to be operating at levels that would disturb these delicate systems. So the paradigm I want to show you today, and this is from a recent article in the Journal of Nuclear Medicine, which I'm sure you'll all wake up and read in the morning, is thinking about this in the paradigm of selecting therapy. So if we're really trying to identify individualized treatment for patients and to be able to choose a therapy based upon the characteristics of the patient's tumor and her makeup, I want to ask ourselves a few questions. Number one, is the target present? If we're giving targeted therapy and the target's not present, we better go look for a different target. Number two, if that target is present and we selected a particular therapy, is that therapy reaching the target in sufficient dose? This is something uniquely, that molecular imaging is uniquely capable of, and actually rather important, and I'll show you a couple of examples. Now, if that's the case that it's not happening, maybe you need to switch the drug or switch the dose. But if everything there is going along okay, the last thing you want to know is, this is targeted therapy. The target's present, the drug's there, but did it actually do something to the tumor? If the answer is yes, then let's go on and continue the treatment. But if we knew early on that the intermediary machinery is not in place and this targeted therapy hasn't done anything to the tumor, we could stop there and go back and try something else. And so there's these three questions that these tools can ask you, and I'm going to show you that example, those examples tonight. The other thing that's very important is fit for purpose. For a while, we have used relatively non-specific compounds that have broad specificity, including glucose metabolism, which happens in a lot of different tumors. And what I want to show you tonight is that when you pick the right molecule and the right target, you get some rather exquisite results. So let's talk a little bit about imaging to guide hormone receptor targeted therapy. Now, the breast oncologist in the room will have this as old hat. For those that aren't breast oncologists, the estrogen receptor is what mediates the response to estrogens and is very important, especially in female physiology in the breast and the uterus and the reproductive cycle. It is a receptor that lives in the nucleus, and it binds to the ligand estradiol, or oestradiol if you're British. And when it binds, it brings the receptors together and brings them to the right spot on the DNA that transcribe a program that can be somewhat specific. In the breast, this is usually related to the process of what the breast does, of making milk, and causes everything from cell proliferation, an increase in vascularity, and an increase in metabolism in the process. And those are all things that tumors need and like to do. And so in many ways, the tumors have hijacked this fundamental breast machinery to help promote their growth. you can interrupt this process in one of two ways. One is you can block the receptor. One of the most well-known drugs for that is tamoxifen, which keeps the ligand from binding to the receptor and prevents this program from happening. Or you can actually reduce the ligand using something called an aromatase inhibitor, which is one of the most popular compounds. But through either of those strategies, you need to know that the target's present. And right now, for the most part, we make that through biopsy determination and special stains under the microscope. And if the estrogen receptor is present, you have a decent chance of responding, up to 75%. But if the estrogen receptor is absent, you have less than 5%. So this is one of the most important diagnoses that come at the time of breast cancer is detected, is the presence or absence of the estrogen receptor, which will very much determine the approach to treatment. All right, so we're going to start back in my paradigm and talk about the predictive marker. Is that estrogen receptor present? And in fact, there is a radiopharmaceutical that's labeled with F18 that looks almost exactly like normal estrogen, estradiol, with a substitution in the 16-alpha position that preserves its properties so that it has very similar binding characteristics to the estrogen receptor, to the native substance. Now, when you take one of these imaging agents, you can't just assume you're getting what you expect, because it's a very complicated process of injecting the radiopharmaceutical, having it move through the body, get metabolized. And so the first thing we have to do is compare what we see on the image to what we know to be our reference standard. And so when this was done back in the 80s, it was compared against an older technique called radioligand binding. Our imaging is, in essence, in vivo radioligand binding this target to the this radiopharmaceutical to the target, showing a good correlation. In work that came from here, Linnell Peterson is going to get cited a lot. Linnell, where are you? Yes, you'll see her name a lot in this process. This was redone in the more modern immune-based approach of immunohistochemistry. But we see that we are measuring what we think it is, and these were very early and important topics. We can also actually see these pictures, and so here are two examples. I'm doing cuts through the body through the coronal plane, so you're looking at the process from the front. You see here a glucose scan, which tells you where active molecules are. There's some normal uptake in the heart. There's some normal excretion of this tracer into the bladder. But there are some spots in the middle of the chest that are lymph nodes that are not supposed to be there. If we go over to the estradiol binding, the fluorestradiol, we can see that all of those sites, and maybe even a couple we couldn't see on the glucose scan, are all binding the estradiol. So they have the target, and that target is present, and we can correlate that with its quantitative uptake. We also see this is a heavily metabolized compound in the liver and goes through the circulation. On the right side is the patient who had a very similar picture, and her biopsy was ER negative. So this tumor, which started out having estrogen receptors, lost it on the way to metastasizing and mutating. And you can see that there's no uptake in these sites, suggesting that this target has been lost. Very important information if you're going to try to treat these patients. And down here, in the kind of imagination that only a nuclear medicine physician can have, that's actually the uterus. And the reason that's important is the uterus has a high concentration of estrogen receptors, so there wasn't something wrong with the technique. We have a positive internal control, and we have uptake, which is very important. So you can measure differences, you can see differences. Now, very importantly, to move this from these early experiments into the clinic, higher level of evidence. In the Asan Medical Center, folks in the Asan Medical Center in Seoul, Korea, did a prospective study that was a level 1 evidence that demonstrated the ability to identify through imaging estrogen receptor positive or estrogen receptor negative breast cancers in a way that met the FDA standards. And so that was part of the evidence, along with a lot of evidence coming from this institution, that supported the approval of this compound of Cerianna. Its inventor, John Katzenellenbogen, one of the most beloved chemists in the field, had been working on this since the 70s, and it got approved about a year ago. But it's now made it into the clinics, and I think many of us are using this routinely. So why do this? We went through all this trouble to get an imaging approach to measure the estrogen receptor that we can do in tissue. And as one of our former colleagues once said to me when we're starting this, he goes, Dave, this is crazy. I can spend $100 and get an estrogen receptor test on a biopsy. Why spend thousands of dollars on a PET test to do this? And the answer is not to replace the biopsy, but to get the ability to assess the full burden of disease in all of the different locations. So I'm going to show you two patients. Again, I'm showing you these coronal images going from head to base. And in the first one, you see an area of increased uptake in the breastbone, which is one of the places breast cancer can spread. On the bottom, we see a patient who has had multiple areas where the arrows are, where it spread to the spine. So this is a pretty common pattern of spread in estrogen receptor positive tumors. Now, that baseline FES study at the top shows that the estrogen receptors are preserved in the bone. Bone is a particularly difficult place to biopsy, a particularly difficult place to assay for the estrogen receptor. And so that presence that you can image is quite helpful. On the bottom, again, here's another one of those tumors that on the way from being estrogen receptor positive to spreading to the bone had some sort of mutation or event that lost the estrogen receptor. Now, we weren't using these at the time to make this determination, and this comes from a paper that Helen Memorial published in the Journal of Clinical Oncology a number of years ago. But this predicted response. The patient on the top had an excellent and sustained response, and you can see that with an early decline in glucose metabolism. The patient on the bottom had multiple different tries of hormonal therapy and eventually had to go on to chemotherapy because the target was missing. Two patients with an identical presentation, two patients who, if they have bone disease in the presence of an estrogen receptor positive tumor, are both going to get hormonal therapy, both with very different responses. Up front, you could have identified that the patient had a chance of responding. In the bottom, now we can actually save that patient a lot of failed rounds of therapy knowing that the target's not present. Now, I mentioned before that this is not just whether the target is there or not by pretty pictures, but how much is there. And these are data that came from pioneering work of Joanne Mortimer and Farrokh Dadashti at WashU, and then about 5 years later, in a slightly different population with a slightly different set of drugs coming from here, from the UW and the Fred Hutch. Now, what these diagrams show is I'm plotting the amount of radiotracer on the y-axis and comparing responders versus non-responders. And if you look at them, there's a lot of overlap. So people looked and said, this thing doesn't predict anything. But this thing does exactly what we would expect. Notice that in both of these methodologies, with actually rather different approaches and rather different patients and rather different drugs, the threshold of this number called an SUV, it's not a Ford Explorer, it's actually a standardized uptake value measure of PET, held across both of these and seems to be the PET equivalent of that pedestal for being ER positive. And in fact, that marker has held more or less in prospective studies to show that without having at least that much uptake in your tumor, patients were very unlikely to respond. So you now take these patients whose breast tumors have spread to a variety of locations, patients very much in a case where good effective endocrine therapy can be very helpful, treat the tumor effectively and be well tolerated. This is a tool that can assess that disease burden across the entirety of the patient, even if you have trouble biopsying one or all of those slots. This is a study that is now making its way through and almost finished in the National Trials Organization, ECOG-ACRIN, again from data that emanated from this institution. Helen Memorial is one of the directors along with Farrokh Dadashti, and this will be completed and look in early stage breast cancer soon to validate this predictive biomarker. Now, there's been a couple of groups in Europe that have also worked in this area, and one of the important recent advances in the field is that in addition to estrogen receptor therapy that is targeted to the estrogen receptor, there are some associated pathways that can be an escape phenomenon that even if you block the estrogen receptor, tumors can still grow and proliferate. One of those is a cell cycle pathway, and a very important class of drugs, such as palbociclib, are CDK4/6 inhibitors that I'm going to show you a little bit later in the talk, that are an important class of adjuncts or helpers or combination therapy for endocrine therapy. And in this very nice paper that came from Europe, they showed that these drugs still require the estrogen target to be able to work. And when there's heterogeneity of the estrogen receptor, when there's less than 100% expression across these very complex diseases, patients are less likely to respond than when they don't. So not only does this work for pure endocrine therapy, but this seems to also work for combined therapy. And we'll get more information on this, including the ECOG-ACRIN trial that I showed you. I will close this section of this by saying this is not the only steroid we can image. The progesterone receptor is a very closely related receptor to the estrogen receptor. I think it's going to be studied here among other places. And the androgen receptor, which is an important marker for prostate cancer, has also been imaged by PET as well. Okay, let's go on to step 2. We got the target. Does the drug reach the target, and does it reach in sufficient quantity to be able to make this work? And so again, I'm going to come back to data that came from here, again from Dr. Linden, and start by looking at images pre and post a blocking drug. Now, when we give this tracer, we give it in minute quantities, and when we flood the system with a pharmacologic level of a blocker, we pretty much occupy all those sites and force the tracer away. And so with tamoxifen, we can see that we have complete blockade of each one of those sites, even though they haven't gone away yet, they have just had their receptors occupied by the drug. Exactly what we like to see. We have the right dose of the drug. Now, fulvestrant came along after tamoxifen and in certain ways was supposed to be an even more potent and powerful drug, but its early implementation in humans was not as wonderful as the mice would have led us to believe. And in fact, this was one of the first pieces of evidence that showed that we were underdosing many patients in this case, that the mice doses, maybe not surprisingly, didn't translate to humans, and using this kind of imaging study, you could see that you had incomplete blockade of that receptor. And about the same time, a trial that doubled the dose, in essence, showed increase in efficacy, and this has actually now become a mainstay of this treatment. Again, the same folks in Europe and the Netherlands did a follow-up study on this and showed that, again, the quantitative level of blockade was an indication of response. And so the blue bars here are responders and the red are not responders. And you can see that if you have the close to complete blockade of the receptor, you're much more likely to respond. And if you haven't completely blocked the receptor and the estrogens can still stimulate that tumor, you have a much lower level of response. Now, the drug companies caught on to this, and as they tried to make the next version of fulvestrant, they go, we don't want to have the wrong dose, and we don't want to necessarily assume that our mouse model is going to work. And so this has actually become a very important tool for drug development. This is from a multi-center trial that involves Memorial Sloan Kettering, Vanderbilt, and Harvard, and showed that you had complete blockade of this drug and the proper dose had been selected. And pretty much any one of these drugs that gets developed in this class now has a small PET scanning study to make sure that the dosing is proper. Again, in data that comes from here, there are other ways of looking at the target. This was a study of combined therapy between endocrine therapy and HDAC inhibitors that are an epigenetically targeted approach that the thought was if you reawaken the estrogen receptors maybe and re-express the estrogen receptors, maybe the drug would work better. We didn't really know how it worked. And this study, which made the cover of that Journal of Nuclear Medicine, you see the name Peterson again, showed a very important finding. These drugs did not re-express the estrogen receptors when they were given. We give the HDAC inhibitors, but really there was no patient that was missing the receptor that had the receptor afterwards. But that said, if the receptor was present, many of those patients went on to have a response to the combined therapy. Again, demonstrating the unique ability of this tool to look at combination therapy where you really don't know what each individual piece of the combination is doing. PET allows you to do that. Okay, has there been a response? So we've got the right dose, we got the right target, we got the right dose. Do we have the right drug? Do we have the right process? Is it working? All right. Now, you didn't know you were going to have to do this, but today you have to be nuclear medicine physicians. There are a couple in the crowd, so they have to hold silent on this. All right, here's a patient that has had 3 months of treatment, pre and post therapy. I will tell you that this bone scan, the hot stuff is abnormal. This is a person with fairly widespread disease. And you have to decide, did this scan get better or did it get worse? Because it makes a difference in what we're going to do for treatment. Okay, so who in the crowd thinks this got better? Who in the crowd thinks this got worse? Who in the crowd thinks, oh my God, I am so glad I'm not a nuclear medicine physician? All right, here's FDG PET. So we move from a bone repair mechanism, which is the way the bone scan works, a bone building mechanism, to this case, the abnormal glucose metabolism that was there. Who thinks this worked? And who now thinks, God, it's easy to be a nuclear medicine physician? I'm going to go get me some of that. So this, again, is data that comes from here. This is work led by Dr. Specht herself, has the name Peterson on it again. And this is data that shows that in a very difficult situation where it really is that hard to tell whether or not these patients respond, the serial FDG PET, the serial glucose metabolism PET, is a predictor of progression-free survival, how long the patient has a response before the disease starts to progress again, whether or not the patient has a skeletally related event, such as a fracture, and starts to hint at overall survival. At Penn, we took this data and said, can we move the clock back a little bit? This was at baseline and after 3 months of therapy. We're showing that you can actually see this profound change in the level of uptake. It actually doesn't look any different on the CT scan, but this profound change in the level of uptake as early as 4 weeks. And so can we get an idea whether or not treatment is working that early? The answer is, I think, yes, but we're actually now studying that in a study that is chaired, again, in ECOG Akron by Dr. Specht and Heather Jacine at the Dana-Farber, called FEATURE. And Jen, I don't quite know how you got FEATURE out of this, but it sounds cool anyway. And this has an early assessment response using PET and actually a circulating DNA marker to go along with it. So we're very excited to see how that trial turns out. Now, it's interesting what the formatting did to my arrows. Now, one of the things was talking about fit for purpose, and we measured glucose metabolism, an indication of the cellular processes and perhaps of viable tumor cells, but we know that there are many steps that happen when a tumor responds. The very last thing that happens is the tumor shrinks. We have to start killing cells, and the tumor has to involute and shrink. Before that, we know we have a change in viable cell number, but even before that, we know that we either have to decrease the cell proliferation, the rate at which these cells are growing, or increase the cell death. And in certain ways, we know some of our approaches are cytostatic. They will stop the tumor. They may not kill the tumor, but they'll keep the tumor from growing. We were talking about this when the Golde family and I were talking this afternoon. That's an appropriate goal. If the patient's doing okay with the tumor and it's not growing and progressing, that's just fine. And so in work that was developed at the University of Washington, and Ken Cronin and Janet Erie are in the audience that really helped move this forward, there was the idea that we could look at DNA synthesis as a much more cogent and earlier marker of this process. And so here is, again, the basic biochemistry. For those who remember it, there are 4 fundamental building blocks of DNA. Thymidine is the only one, 4 of them, that is used only for cell proliferation and not RNA, so a very specific marker of DNA synthesis, and the cell has to synthesize DNA to be able to reproduce itself. And you can see that there, again, is a very close analogy between thymidine itself, the native molecule, and this fluorine-18 labeled molecule. So how well does this work? And here are 3 different examples to show how when you have a fit-for-purpose molecule that looks at early response, how well this can work. The first one is from an Akron study published a few years ago that showed after a single dose of chemotherapy, within 7 days, we could see declines in therapy in successful treatment. So again, can we tell whether or not something's working after 7 days? Maybe, and maybe we can use this to guide therapy. In another study that came from University of Washington, the SCCA, and the Fred Hutch, we were able to show that this was an early indication of response to endocrine therapy, to hormone therapy. This had been done in tissue by a number of studies that show that you could measure proliferation as an early indication of the response. This confirms that you can do it with this FLT, this proliferation tracer. And then finally, in work that we have done at the University of Pennsylvania that has at least been presented in abstract form, we are taking these CDK4/6 inhibitors, these cell cycle inhibitors that control the proliferation of the tumor, and we're doing something unusual. It's actually almost like an old biology experiment. We're pulsing the tumors. We're turning them off, and we're turning them back on so they all come within sync. And as they start to proliferate again, and they're all in sync, that is the optimal time to be able to kill the tumors with chemotherapy. But one of the things is we had no idea whether or not we were dosing and what the timing of the pulsing is. And one of the most extraordinary things that I've seen since working in this field, this is after 3 days of these pills that do the cell cycle inhibition, we see almost complete shutdown in those patients that have a good response. And if I showed you the other picture that repulsed that, that withdrew that afterward to get these synchronized, the level actually came back a little higher. So it didn't happen in every patient, but when we got the dosing and timing right in the right patient, it was a unique opportunity to show in this combination therapy that we were doing exactly what this drug was designed to do, which was to stop cell proliferation. So it's all about the fit for purpose. And some other exciting work, the group at the University of Washington University, Farouk Dadashi, Joanne Mortimer, had used glucose metabolism to look at this unusual property of tamoxifen. Tamoxifen actually stimulates through the estrogen receptor before it starts blocking. And women taking tamoxifen, they get a flare in their symptoms, is often an indication, an early flare, that it's working. So this was an FDG flare, a flare in glucose metabolism that showed if you had that flare within 1 week of treatment, you were much more likely to respond than if your tumor basically just ignored it. And again, in very exciting data, there have been a recent study showing that imaging the progesterone receptor is an even more powerful way of showing that your estrogen receptor is working and that blocking it is likely to lead to a treatment. This is a recent paper from Nature Communications that supports this pharmacodynamic marker, again, rather than using a broad marker like glucose, something that's very specific to what you're looking at. And it comes back again to this fit for purpose idea and the idea that we should have markers that will give us very specific ideas of what's happening in these different targeted drugs. All right, I'm going to switch gears now to PARP therapy. I'm going to show you some of our early work here. Again, I want to remind you I do have a conflict of interest as I have commercialization going on. All right, so for those that are not DNA repair experts, and I am an erstwhile one that learned this as part of the process, there are a couple of major forms of DNA repair, which is essential to life. DNA gets damaged in the process of life, gets damaged in certain treatments, and its repair is critical to normal function, and its repair is actually not what you want when you're trying to treat and damage and kill a tumor. So one of the two forms of repair is non-homologous DNA repair, basically not necessarily using the other strand to do that. And this is an important molecule here is a molecule called PARP. And a number of years ago, there were inhibitors that bind to PARP and prevent its action and actually have the PARP get stuck in the DNA. And in the absence of the other major form of repair, which is homologous repair, this would lead to trapping and further DNA damage and cell death. And that homologous repair is part of what BRCA genes do. And so this is very important in patients who have mutations in their tumor of BRCA and related genes, and this has become a very effective therapy. And it works, as I mentioned, through this process of synthetic lethality, where if you have these two methods of being able to do DNA repair, and you have on one side let me make sure I got this right. Oh, sorry. You have on one side the inability to do homologous repair. Blocking the PARP repair results in very selective and significant synthetic lethality, and these have now become an important part of breast and other BRCA-related cancers. Now, the way in which this is chosen right now, for the most part, is a genomic assay. So this is a somewhat unique approach in that the biomarker for deciding whether or not to use this drug is all based upon genes and looking at the genes that are involved in DNA repair. And as what I've shown you today is that the ability to measure the target and the place where the drug binds can be quite important. And so Bob Mack, who directs our radiochemistry, developed a radio tracer that is an analog to PARP inhibitors and binds to the same target called fluorothanatrase, or FTT, and when labeled with F18 can actually image that target to where the drug binds, much as what I showed you with the estrogen receptor. We also have the ability to label this with a different isotope and use it for staining in the tissue, so we can actually do tissue assays for comparison. Now, the very first paper in this came from Delphine Chen, who's also sitting in the audience, before Bob Mack moved to Penn and Delphine moved to here from Washington University. and had some important studies. Number one is showed on the left side of your diagram in mouse models that when you image these mouse models and gave them a blocker, you reduce the level of uptake. That's the same thing you saw before. If we give a drug, we should be able to start occupying that target. And when she gave something, olaparib is one of the PARP inhibitors, when she gave kind of a fake PARP inhibitor of aniparib, did not block it. So it showed the specificity of the target. The other important thing was to show that the biodistribution of this target, its normal organ appearance, was actually pretty favorable and was a reasonable way to be able to image many of the places that breast and other cancers would go. And in this early paper, Delphine demonstrated, for example, in this liver cancer, cholangiocarcinoma, that you could see uptake of PARP that correlated with what was expected. About the same time when Bob Mack moved to Penn, we started to work on this focusing on breast cancer. We actually started this in targeted therapy. You will note there was a man cough in the paper. It was my daughter working with Bob's lab at the time. And then this was part of the early work that then motivated Christine Edmonds, who trained at Penn, is now back at Penn, to do one of the earliest papers looking at the correlation of this molecule's binding in tissue with what we would measure by sampling that tissue in this mouse models. And again, we're doing these imaging studies to look for targets. We got to validate that we're measuring what we think we are. And what you can see is that in different mouse models that had varying levels of PARP expression, we saw a very good correlation. We then had a parallel study going on in ovarian cancer, another important BRCA-related tumor, and a tumor where PARP inhibitors are used quite heavily. And we were able to show variable level of expression in these tumors, suggesting that this might add information on what was happening. You may have the genomic scar that leads to the synthetic lethality, but if the target's not present, based upon some studies that we've done, mostly in the preclinical realm at this point, you wouldn't expect the drug to work. If there's nothing for the drug to bind to, the chance that it's going to work is small. And again, in this early study, we went back and did what we always do to validate that what we're measuring quantitatively on a PET scan matches what we see in the tissue. It sounds a little bit like a broken record, but it's a very important part of the validation that we do. We're actually now in a 3-center trial with Penn, MD Anderson, and WashU to repeat the study in a multi-center trial with the idea of trying to move this in the direction of FDA approval. Now, shortly later, Elizabeth McDonald at our place, who also trained here before she moved to Penn in breast imaging, put together a paper that was published in JAMA Oncology that looked at primary breast cancers across a spectrum of diseases and characterized this based upon their phenotype, whether or not they expressed estrogen receptors, whether or not they expressed HER2, whether or not they were triple-negative breast cancer. Many of you have heard of these terms. And what she saw was tremendous variation in PARP expression across those. Now, interestingly, where you see a little letter, every one of those little letters is a recognized BRCA-related mutation. And this is how we choose these targets right now. But the variability of PARP expression, including some low PARP expression in these tumors that are identified on the basis of mutation, was about the same as what it was across these different phenotypes. And so this suggested, and we're just at the beginning, we're just going to present some of the very first results of this at the upcoming Society of Nuclear Medicine meeting. This suggested that this may be a marker that may be complementary in trying to identify and help predict beyond the genomics who might respond and might not. So stay tuned, more coming, but it looks promising so far. The other thing that Elizabeth did was to take a handful of patients. We've got these studies going on in both breast and ovarian cancer, image them before and after the PARP inhibitor. So if you're dosing the PARP inhibitor right, you have this pharmacologic level of a drug that's binding to the same target as your tiny little bit of PET tracer, and you should see that blockade. And in this paper, which is largely anecdotal, it's just examples so far, we can see that in the patient that had a block from high uptake to no uptake, she had a good response to the PARP inhibitor, whereas the patient that didn't have much of the target to begin with and had not much change with treatment that you see on your right did not. So again, early data suggesting that this may be a way to help predict who's likely to respond to these PARP inhibitors. So one way we might think about using this is to take this PARP platform, and when we look on the light bulb and try to identify whether or not these tumors are binding to it, is to use this much as we are now doing with the estrogen receptor tracer to decide in patients who have PARP-resistant tumors, this is a very important problem in ovarian cancer, whether or not to think about a combination therapy or a different PARP, or whether if the target is missing to think about alternative therapy. Another very exciting approach is illustrated in, before I get there, is coming in a second. But one of the things that we found is in some of the patients we studied so far in ovarian cancer who had lots of PARP expression to begin with that blocked down after the drug, had a response to the drug for a while and then became resistant. And when they became resistant, they actually preserved their PARP expression. The disease that came back afterwards, even though it had escaped that drug, still expressed these high levels of PARP that could be blocked with the drug. So this suggests the possibility of going beyond simply predicting drug therapy and thinking about alternative approaches. So if I go back to this scenario where we have developed this ability to image this particular tumor, I can change isotopes, and I'm going to change this to something that's actually made here and also at Penn, an alpha emitter called astatine, and parthenotrace, which is a very similar molecule that binds to the same site, and we can give the heat deer, the lightning bolt, as very targeted radiotherapy that moves in this direction. Now, why would somebody want to do that, and why would this be an advantage process? Well, there are a couple of reasons for thinking this could be helpful. It's fit for purpose. This is identifying a particular target that is associated with a particular group of tumors. But very importantly, this is localized in the right place. These alpha emitters are a very interesting form of radiation that deliver potent radiation over a short space. And they deposit almost all their energy within a very microscopic travel point from where they're emitted. So if you're going to try to kill cells, you want to break both of their strands of DNA. And if you wanted to do that, you want that with something that's sitting on the DNA in the nucleus, which is where the PARP is located. And what this graph shows you, if you just take naked astatine, which looks a little bit like iodine, and throw it on the cell, and then actually put that astatine at the same concentration in this molecule that brings it to the nucleus, you have greater than 100% increase in potency. So now a very targeted approach. Now, we have studied this first at Penn, and this is a paper from Ron McVondy, in a neuroblastoma model. This comes from a collaboration between Dan Prima and John Marissa, a neuroblastoma expert at Penn, who looked at mouse models and found that this particular drug could have significant potency at slowing the growth of tumors. And so from those initial studies, they went into a mouse model where they gave much higher doses. And they saw something that hadn't been seen in this model before, and that is complete control of this tumor out to 100 days. Now, on the right side, if you're used to looking at survival curves, you'll notice that that curve for the mouse that had a single high dose didn't do too well. They had enough marrow toxicity that mouse died. But as these doses were fractionated, we're used to giving these doses not all at once, but in fractions, the normal marrow got a chance to repair, but the tumor never did. And if anything, when the tumor got radiated, what did it do to try to repair itself? It expressed more PARP. And so this is, in essence, a self-amplifying process that can be somewhat selective. And so the vast majority of these mice survived out to 100 days, which hadn't been seen before. And so this is something we're very excited about. We're thinking about pivoting this back to ovarian cancer. Taking this into adults, and is a very exciting way of thinking about leveraging this compound for not only a diagnostic, but a theranostic or a therapeutic. All right, I'm going to stop by just showing you a couple of tools that are kind of fun. Those that want to come to my session tomorrow at the breast retreat will see a little bit more of this. One of the things that we are one of two centers in the U.S. that has been able to do is make a whole body PET scanner. When you make a whole body PET scanner, you pretty much can detect close to every molecule or a high fraction of the molecules that are injected and come out of the body. So you can image full body processes at once and look at dynamic approaches. You also can use very low levels of radioactivity and still get very high quality pictures. And this is actually a home-built device. This is the lab where I did my PhD that have now constructed one of the world's only two scanners. They're now being commercialized, and there are more of them around. And what this enabled us to do was to look at two different fuels, glutamine and glucose. If you come tomorrow, I'll tell you more about that. But to be able to do this in a single injection in the same patient, to be able to look at two tracers in the same patient and get full characterization of their dynamic curves. And so we're very excited about this. This is something that was a Cancer Moonshot grant to develop this technique. And we think this has widespread activation and actually leverages a paper that Dr. Krohn published a number of years ago showing you how much better two tracers are than any one single tracer. So we're very excited to take this into practice. Last but not least, we've taken that same instrumentation capabilities at our place and put a breast-dedicated imaging device. That's also going on in development here. But we also put it on the same device as a breast tomosynthesis, a modern mammography approach. And so we're just about to start trials. We're actually going to use our estrogen receptor tracer to see if this combination can identify the extent of spread within the breast, which is actually a real challenge sometimes in the estrogen receptor positive patients. So we got a couple of new gadgets that will bring new tools for us for breast cancer from the whole body all the way down to the breast. And so it was fun for me because I got to return to my roots as an instrumentation geek, so to speak, but be able to now take advantage of these new applications and important things that we can do to help patients. So with that, I'm going to stop and tell you that hopefully I've shown you that PET molecular imaging can measure targets, measure the drug binding engagement to help with dosing, to look for a specific response to the drugs, and that there's strong evidence that this is useful for the estrogen receptor, in fact, so strong that it's been FDA approved. We're very excited about other targets, including early work on the DNA damage targeted agent PARP, following a very similar paradigm to what we had done for estradiol. And there's active research going on in a whole bunch of other targets that are likely to provide this very individualized approach to breast cancer, and may one day also identify very individualized and localized radiotherapy using therapeutic radioisotopes. So with that, I want to thank sponsors and grants coming from NIH, the Penn Vassar Center, the Rifkin Center, based in Seattle, helped support some of the ovarian cancer work we did. And I want to thank my colleagues at the University of Pennsylvania, many colleagues who contributed to the work. This is just a subset of them at the University of Washington or Fred Hutch and Fred Hutch and the University of Washington, and many other folks who come from the same diaspora or come from similar roots. And with that, I will stop and be happy to take questions.
So we're monitoring the chat, and Dr. Flanagan astutely noted that the liver looked really hot in those studies. So what can you? Her question was, can you use FES to detect liver mets?
Yeah. So just to paraphrase the question with a little bit more detail, in those first set of studies I showed you, the brightest thing was the liver. These estrogens are a class of drugs called steroids, not to build up muscles for weightlifters, but these steroids, no matter what flavor they're in, get heavily metabolized in the liver. And so one of the weak spots of the estrogen receptor tracers, the progesterone receptor tracers, that are also steroid receptors, is it's very hard to visualize the image. And so there's got to be some work done to be able to visualize the image, which is an important part of metastases. One of the things we're getting started on with our whole body PET scanners is to take delayed images, because the normal liver will wash out and be retained in the liver. But short of that, I think that liver hotspot is going to be one of the blind spots of that particular tracer.
Other questions? Iris.
Really nice talk. So the home-built system that you guys have for whole body PET imaging, does it overcome resolution images that are, remain to PET? Can you go beyond 0.5 millimeters, or do you see it advancing so that you can go beyond that limitation? Yeah.
So this is a debate that I remember I was one of Harold Varmus's important question studies, and this debate came up on that question. These devices will improve the quality of images and allow you to distinguish smaller amounts of activity, mostly because you have such good statistics. You're getting so much information, you can do that. And in theory, you may be able to use amplification techniques to sharpen those images. But these techniques will allow you to look at probably about 4 or 5 millimeter resolution with good high statistics and important great molecular fidelity. But I don't think we're going to get much below that unless we go to very small devices and maybe start to get down to 1 millimeter or 2 in a dedicated breast device. So the strength of these modalities is never going to be the ability to get down to that microscopic level because of the nature of what we do. The strength of them is really to be able to get in vivo imaging down to a level of sort of semi-macroscopic or macroscopic tumor size. Interestingly enough, there have been some radiomics applied to this to try to dig some of that information out. We've been able to leverage this 4-dimensional whole body data and start to tease some of those features out because there's a lot of molecular fidelity. But I'm afraid we're stuck with nature and physics to the point where we're probably not going to get down to the single cell level here, unfortunately.
Do you think the whole body will get you over the GI problem of nonspecific uptake there?
Yeah, so there are always going to be a certain amount of nonspecific uptakes in this, and the ability to look over time allows you to look for tracer that is retained in the space that you see and see it move through the system in other areas like the bowel and the kidneys. So I do think that's going to help. I don't know that it's going to necessarily solve that problem, but the ability to look at either long-term or short-term dynamic studies, I think, are going to improve the quality and distinction of what we can look at.
Okay, we want to try to go with the hand raise here.
Okay.
Somebody, Marianne Duvar-Gall has her hand raised, so let's see what we can do.
He's on mute.
Yeah, can you unmute yourself? No. You can't. Can I unmute you?
Maybe have to type it in the chat.
You might have to go with the chat.
Yeah.
Oh, it works. Can you hear me?
There you go. Yeah, okay. Yes.
So do you see using the PARP mimicking tracer as a predictor whether PARP would work in patients before we even give them a PARP? Maybe I missed that part of your presentation.
Yeah, I went pretty fast by the end, but in analogy to what we saw with the estrogen receptor, you might think of that. Now, I want to acknowledge this is a stretch. We've really not been looking at this target up till now, and I'm not sure we've had the tools to do it. And I think it is necessary to bind the tracer for the drug to work. It's not the only thing that would be there. And so one of the ideas is that if you had lower absent PARP expression and binding of the drug, even if you had the right BRCA-related genes, you might not respond, or you may at least need some of that information for drug dosing. What's a little different about the PARP inhibitor is this really intriguing idea in the breast cancer data that I showed you is there are a lot of high PARP expressing tumors out there that don't have a gene that we see. That doesn't guarantee they're going to work. They still need the genetic defect to do that. But I think it opens up the idea that there may be additional patients out there who might benefit from the right PARP inhibitor that will take a lot more work to support that hypothesis, but opens up the exciting possibility that it's not just that PIT tells you who you can't treat, like it is from the estrogen receptor, but may identify patients who you can treat that we didn't know about. You know, for all the DNA damage people listening, I know we have a lot to do to prove that point, and that's a very simplistic idea, but I think it's an intriguing process to have the target imaged along with the genomic scar to see how the two work together. Some of our data is showing when you get to combination therapy, not surprisingly, the information that you get out of serial PARP inhibition is quite good. We'll be presenting some of this at the Society of Nuclear Medicine and some mouse models.
And one more question for you. So, you talked about a whole bunch of different tracers, proliferation. You didn't talk about PSMA, but you talked about estrogen, progesterone. And this PARP. But, what I guess in our dreams is that we're going to be able to define these pathways for every patient. So could you just chat a little bit about androgen and about HER2?
Yes. So one of the things that I think was part of the philosophy of the group that we had here, you could in principle label every new drug with a PET agent and have this at your armamentarium, you call down your cyclotron and say, hey, give me a little bit of flavor A and a little bit of flavor B. In fact, there's often 10, 20 years worth of work in validating that. So the idea is to get high prevalence targets. Two of those include the ones you're talking about. There is work showing with HER2 imaging agents something very similar to what we saw with FES, showing the presence or the absence of the target. That has been shown with basically a labeled antibody, which may not be the optimal way to do that imaging, and there's some newer targets coming along. The androgen receptor has also been looked at in prostate cancer. There's been imaging work that's gone on at Sloan Kettering and Washington University in St. Louis. It's a little bit more ubiquitously expressed receptor and is lost a little bit less frequently. And again, there's people that know a lot more about prostate that are in the room and listening to me. But it hasn't caught on quite to the same extent that the estrogen receptor does, where the presence of a large class of tumors that are missing that estrogen receptor or lose the estrogen receptor, I think has the potential to impact therapy. But there's more of these targets coming. I only went through a few. There's very exciting work going on in immune-specific targeting that looks at immunotherapy activation.
I'm not a scientist, so this will be more of a layperson question. But I was just curious, you talked about how this work might have some applications for prostate cancer or ovarian cancer, and of course with breast cancer. How does that inform the likelihood of bringing therapeutics to market? Is that helpful? Is that competitive? Can you talk a little bit about that?
Yeah, that's a great question. And I think all of us that have worked in this area with PET have struggled with it a little bit. In theory, this should be something that should be an intimate and an important part of drug development, because you develop this drug and you're going from mice and animals into humans. This really informs you what happens in vivo. And as a matter of fact, those drug companies that are developing a psychiatric drug wouldn't even put that drug into humans without doing a PET validation of that. That has been a little slower to catch on in oncology, and it's been very difficult to say, wow, if I'm developed this drug all my life, I'm going to be damned if some imaging study is going to kill my drug. And so it's actually a very interesting pharmacologic and industry sociology. Again, I'm excited about FES as the drug companies caught on that if they use this, they're going to get the dosing of their new estrogen blockers correct. And so I think that's an early indication that this could not only help clinical patients, but could be very important tools for drug development. Again, the hard part is the effort that it takes to develop a radiopharmaceutical is significant. In some cases, almost as much as what it would take to do the drug. So you'd like to have this at the time you start investigating the drug, but if the drug target doesn't turn out to be worthwhile, you've wasted all that time on the PET tracer. So there's a delicate balance between these processes, but I'm a fan and an advocate that this could really help in not only directing early drug development, but targeting to the right patients. You know, if trastuzumab or Herceptin had been tried and we didn't have a test for HER2, that drug would have failed. And so we have some tools to better identify the patients likely to benefit from these, and that I think will help with the drug testing and approval process. Good question. Thank you.
Good question. Any other questions? All right, you can hear you're up. Excellent.
Well, first, please join me again in thanking Dr. Mankoff. And on behalf of the Golde family, we really want to thank everyone for coming this evening and for all of you who joined us online. We're going to close by playing a short video. When Helen was in the final weeks of her life, many friends and family visited her. And after my brother's family left Washington, D.C., where my sister lived, Beth, my beloved sister-in-law, and Eric Golde, Helen's beloved nephew, orchestrated and edited and sent this video to her during her treatment. It gave her great joy, and we think it's an upbeat way to end our time together. And I want to note that many of the people who are joining us online are names I recognize and who are sort of implicated in this video. So some of you also in this room may see yourselves. And so following the video, we invite all of you who are here in person to join us right across the hall for a reception. We have food, we have wine, we have the ability to actually hug each other in person, and so we're delighted to be with you. And I'll get out of the way. This lovely person can actually do